For light microscopy, the basic reaction of stains follows the principle of the attraction of opposites. The acidic components of cells stain with a basic stain and are called basophilic, while the basic components of cells stain with an acidic stain. There are a number of different histological stains used for light microscopy. Perhaps the most commonly used stain is hematoxylin and eosin, abbreviated H and E, which is good for visualizing general structure. Hematoxylin is basophilic, staining nuclei blue. Eosin is acidophilic, staining the cytoplasm pink. To visualize connective tissue, a combination of 3 stains is used to visualize the nucleus, cytoplasm, and matrix. Examples of commonly used stains are Masson's trichrome or Mallory's triple CT stain, which will stain the nucleus dark red or purple, the cytoplasm pink, and connective tissue blue. Periodic acid shifts, abbreviated as PAS, detects glycogen in tissue and will stain structures containing a high proportion of carbohydrate macromolecules such as glycogen, glycoproteins, and proteoglycan. PAS typically stains connective tissue, mucus, the glycocalyx, and basal lamina. Silver impregnation is used to trace nerves and to stain Golgi and reticular fibers. Silver is also useful for staining Pseudomonas, Legionella, Leptospira, and H. pylori. Right stain is a histologic stain that facilitates the identification of blood cell types. It is primarily used to stain peripheral blood smears and bone marrow aspirates. Right stain uses azure blue stains to stain white blood cell granules basophilic or neutrophilic and is used with eosin for RBS and eosinophilic. Next, we will take a look at the histology of tissues. In traditional classifications, there are four forms of fibrous tissue: epithelial, connective, muscle, and nervous. In this section, we will explore the histology of these four types of tissue. We begin with simple epithelium. Simple epithelium is one cell thick, where every cell is in contact with the basement membrane. Simple epithelium may be further divided into squamous, cuboidal, columnar, and pseudostratified. Simple squamous epithelial cells have a flat, smooth surface that provides a smooth, low-friction surface. Nuclei tend to be flattened and elliptical due to the flattened shape of the cell. Squamous epithelia are found lining surfaces that utilize passive diffusion, such as the alveolar epithelium in the lungs, as well as lining cavities such as blood vessels and pericardium. As indicated by their name, cuboidal cells appear square or cuboidal in shape. Each cell has a spherical nucleus in the center of the cell. Cuboidal epithelium is typically found in secretive or absorptive tissue, such as the lining of the kidney tubules. Columnar cells are elongated such that they are taller than they are wide, with elongated nuclei that are usually located near the base of the cells. They may or may not contain microvilli or cilia. Columnar epithelium lines the stomach and the intestines, while other columnar cells are specialized for sensory reception in the nose, ears, and taste buds of the tongue. Pseudostratified epithelium is comprised of simple columnar epithelial cells with nuclei at varying heights, giving the impression of stratification when in fact there is a single layer of cells. Pseudostratified cells may possess cilia. These cells are found in the airways, where the cilia beats to remove particles, and in the uterus and fallopian tubes, where the cilia move the ovum to the uterus. Stratified epithelium is multilayered, and is found in body linings where chemical and/or mechanical injury is common, such as the skin. Stratified epithelium may be further specialized to be keratinized, in which the apical layers are dead, anuclear, and contain keratin to create waterproof layer of cells, or transitional, found in tissues that can stretch, such as the bladder. The epidermis, or outer layer of skin, is a stratified squamous epithelium comprised of proliferating basal and differentiated suprabasal keratinocytes, which make up 95% of epidermal content. The layers of the epidermis from its surface to base are stratum corneum, stratum lucidum, stratum granulosum, stratum spinosum, stratum basalis. You can remember this with the mnemonic Californians like girls in string bikinis. Skin color is determined by melanocytes, which are melanin-producing cells localized to the stratum basalis. Cell junctions act to hold together cells, either with each other or between a cell and the extracellular matrix. In vertebrates, the primary forms of cell junctions are tight junctions. These seal neighboring cells together in an epithelial sheet to prevent leakage of molecules between cells. Adherens junction joins an actin bundle in one cell to a similar bundle in a neighboring cell. Desmosome joins the intermediate filaments in one cell to those in neighboring cells. And gap junction forms channels that permit passage of small, The parathyroids are located on the posterior surface of the thyroid gland. Although they are quite small, about the size of a grain of rice, they are easily distinguished in H&E staining, where you may recognize parathyroid chief cells as smaller, numerous, darker staining cells that synthesize PTH, and larger, fewer, lighter staining oxyphil cells of unknown function. You may also see adipocytes, which increase with age. The adrenal gland is divided into the outer cortex and inner medulla. The cortex is further divided into three layers: the zona glomerulosa, which is the primary site for mineralocorticoid production; the zona fasciculata, the primary site for glucocorticoid production; and the zona reticularis, which produces androgens. The medulla contains chromaffin cells that produce epinephrine and norepinephrine. Histological sectioning of the testicular parenchyma reveals a number of important structures, including the lumen of tubulus seminiferus contortus, spermatids, spermatocytes, spermatogonia, Sertoli cells, myofibroblasts, Leydig cells, and capillaries. Sectioning of the uterus reveals 4 layers. From outside in, they are the endometrium, which is a single layer of columnar epithelium on stromal layer of connectal tissue, the myometrium, made up of smooth muscle, the perimetrium, a loose surrounding tissue, and the peritoneum. Ovarian histology varies with the development phase of each follicle. Each follicle contains a single oocyte, from which a single mature ovum is released once per menstrual cycle. Each oocyte is surrounded by granulosa cells within the follicle that produce LH and respond to a number of circulating hormones, including testosterone and FSH. Now, let's turn our attention to the nervous system. The nervous system is made up of a number of different cells that form vast information networks within the body. There are 2 basic types of neural cells, neurons and glia. Neurons are polarized cells with a cell body, or soma, several short extensions off the cell body that receive signals from neighboring neurons called dendrites, and a single long extension called an axon covered with myelin that sends signals to recipient cells. Neurons are directly responsible for transmitting information through a complex interaction of electrical and chemical signals. A bundled group of neurons is called a nerve. Glial cells play a supportive role by providing structural support and insulation to neurons and remove neurotransmitters at the synapse. Glial cells come in a variety of specialized forms, including oligodendrocytes, which produce myelin in the central nervous system, Schwann cells, which produce myelin in the peripheral nervous system, and astrocytes that provide physical support and repair and aid in the maintenance of the blood-brain barrier. Unlike neurons, glia do not transmit electrical signals. When looking at peripheral nerves, there are 3 layers of the peripheral nerves from the outside in. First is the epineurium, which is dense connective tissue that envelops the entire nerve, acting as a protective sheath. Next is the perineurium, a permeability barrier that surrounds a fascicle of nerve fibers. And third is the endoneurium, which surrounds a single nerve fiber. Their prefixes aid in remembering their definitions. Epi means outer, peri means around, and endo means inner. In humans, the olfactory system detects smell through contact of odorants with olfactory receptors located in the olfactory epithelium of the olfactory bulb. The epithelium lies just beneath bone. Activation of olfactory receptors sends signals to the olfactory receptor neurons called mitral cells at synapses called glomeruli. Within the eye, there is an area of specialized cells that require our attention. The retina is the light-sensitive tissue lining the inner surface of the eye, which translates visualization of objects into neural signals. The vertebrate retina has 10 distinct layers, 4 of which contain photoreceptors: the pigment epithelium, the photoreceptor layer, bipolar cells, and the ganglion cell layer. Finally, let's take a look at the structure of the ear. The cochlea is the auditory portion of the inner ear. The sensory organ of hearing is the organ of Corti. The organ of Corti is a sensory epithelium comprised of hair cells topped with stereocilia that regulate hair cell excitability. Sound-induced movement of hair cells triggers signaling to the auditory cortex via spiral ganglion neurons.